of homologous. The diagram below shows the pairing of homologous chromosomes in a cell undergoing meiosis. Okay, and we're showing chiasma formation on a homologous pair. Name the type of cell that undergoes meiosis. Well, that's just a straight KU. That is a gamete mother cell. Explain how the chiasma formation between the paired homologous chromosomes shown in the diagram leads to variation. Okay, so chiasma by itself doesn't do anything. That is just the touch point. But when that forms, what you can get is crossing over. Okay, now that's the most essential thing, that we recognise that it's not the chiasma that actually does the job, it is the crossing over. And if you have crossing over, then you get recombinant chromosomes. So what we've got is, if this is chromosome homologous 1, okay, and I'm just going to do the homologous 2 in a different colour, okay, so these are the same order of alleles, and you've got your centromere in the same place. So let's say on this one, just for ease, okay, this one has this has all little ones, okay, so little e all the way down to little f, and then on the other one we have the big E's, big B, and obviously these are on both sides of the chromosome, because the chromosome there is actually two sister chromatids, exact copies of each other. So if there is a crossover point, so an actual chiasma forms, so say it forms here, okay, now at that point, if this actually snaps and rejoins, one possibility is it snaps and just grabs back hold of what it had originally, so there would be no recombinant organisation of the alleles. But if you actually get it to snap and it crosses over, we end up with, well, I've got a green one there actually, I could use that. Okay, we end up with this. Let me just put, sorry, too many colours. Well, not too many colours, you can never have too many colours. Okay, here, and that one. And what we have now is that when this finally does its full separation, okay, this top one and the bottom one are fully as the parental types were. But this one now has a big A, a big B, a big C, and then it's got a little d, a little e, and a little f. So this is now a mixture of the alleles from mum and dad, if we're looking at this parental wise. And this one's got little e, little b, little c, and now has a big D, a big E, and a big F. That's a little one. And on the other side, obviously, the parental, as I say, would just remain this way. Okay, so these ones here, that's your variation. So you have those 2 points, the crossing over, and then you've got a new combination of alleles. Okay. Name the process that ensures haploid gametes produced by meiosis contain a mixture of chromosomes of maternal and paternal origin. Right, that is independent assortment. So that means if you're looking at a cell, okay, and you've got, let's make it easy, let's go for 3 different chromosomes, okay. So here's a copy from mum for 3 different chromosomes, and let's say that dad's line up like that, okay. Now that would mean if you split this, you would actually end up with 4 gametes, 2 of which were totally maternal DNA and 2 of which were totally paternal DNA. Obviously, just to make things more complicated, you could get crossing over in here, but let's assume there's none. Okay, so that's actually not that likely. It's doable, but let's see that this happened instead. So what you get here is independently each homologous pair lines up, so it doesn't have to go mum dad mum dad mum dad. Okay, it can go mum dad dad mum mum dad. Okay, so it's a completely random chance as to which ones go where, and then you've also got that increased variation in the middle because of recombination. So yeah, that's what we mean by independent assortment. Obviously for us it's super complicated and super potentially increasing variation because we have 23 pairs rather than 3. Okay, and that's that question.